Should I say good morning or good afternoon? I think it's good afternoon. As a matter of introduction, I am I think I've been quite a while in Loma Linda as a student. I was a missionary in Africa. When I came back, I decided to go into the public health. I did a couple of master degrees in epidemiology and global health. And now with Dr. Dos Santos, I'm trying to complete a doctoral program in preventive care. So this is very exciting. And this topic that I'm going to present today has come to me as a vision, because I didn't have an introduction, a complete introduction in this topic before. I read a few things here and there, but I didn't know that it was so profound, so life-changing that I really want to pass it to you. So having said that, let's go into the topic. Let's have a conversation on this issue of the microbiome. Let's look into the. Can you see my?
Yeah.
Okay.
Yes, we can see it.
Can you see that? I have what it says starting subtitles. Can you see that?
No, you have to click more. You have to click more.
Yeah, no.
Oh, you have. Yeah, we have.
Yeah, we can see it.
Oh my goodness, that is an issue that I'm having with my computer. I don't know why it keeps recording what I say, regardless of I turn it off or on the microphone. So if someone knows how to deal with that, I would greatly appreciate. Okay, so let's talk about the topic this morning, the microbiome. The title is A Parallel Universe. So we see something interesting here. Science has taken us to very high levels of technology, and we understand lots of things about our universe, the position of Earth in the planetary system. We know about other stars, galaxies, and we are finding parallel Earths that supposedly one day we will go there and live happily and eternally. But the issue is that just around us and within us, we have another universe. I called it a parallel universe. And this is a universe that we cannot see with our naked eyes. We know it because of its consequential effects on our health, and research is showing us how important it is for our health. And I have several questions of these, the effect of these microbes that live around us and within us. This is what we call the microbiome. And we'll look into some definition to see where exactly are we. So we have been in contact with microbes since the very creation. When God said that he formed man from the dust of the earth. So incorporated the microbes in our body to help us. So with this picture, this picture is saying that when we look at ourselves, for example, in the mirror, we cannot think anymore that that's all we are seeing, because there's another part that is composed for 40 trillion microorganisms that we do not see. And that's very interesting. These microorganisms collectively are called a new organ for us, because they have their own DNA, and this is an organism that we need to learn and to treat, to learn about it and to treat well. But what's interesting here, as because we are in preventive care, is that what we eat may greatly affect these microbes and their response to our bodies in the matter of health and other issues. So what I want to get into next is the following. You see the pictures of these four guys. Initially we didn't know what the bacteria was doing, even though we had several plagues in our history, like the plague that decimated Europe and the northern part of Asia in the 1300s. We didn't know, but it was caused by Yersinia pestis, one of these deadly bacteria. So we have here Mr. Koch or Koch. Hold on a second. My apologies. We have Van Leeuwenhoek. He was able to, in 1618, to create or build a very good quality microscope to see these small organisms. And after that, later on, Mr. Koch described several bacteria, including the Bacillus anthracis. With this, he gave birth to the microbiology and the public health. Later on, Mr. Mednikov, he's an interesting guy. He was a Russian guy, was born in Ukraine, and he had the idea that there must be some good and bad organisms. So he's the father of probiotics. During a cholera epidemic, he decided to drink a cocktail of microbes composed mostly with cholera bacteria. Nothing happened to him. However, he gave it to his assistants. One was okay, but the other one almost died. Since then, he proposed that there is a good bacteria and a bad bacteria in our gut that would be very helpful to our health. So later on in 2001, Mr. Lederberg proposed the name of microbiome to this conglomerate of bacteria existing in our bodies. So that's very interesting to me. In 2003, the Human Genome Project was finished, and the human genome was completed. But the reason I mention this is because this paved the way for other researchers to study another genomes, including several of the genera bacteria. Okay, let's look into some definitions now. We have the term microbiota. Microbiota is the collection, the actual collection of bacteria, viruses, fungi, and other single-cell organisms that live in our bodies. It's called a supportive organ for some reason. Then we have the term microbiome. As we can see here, it's the combined genetic material of the microorganisms in a particular environment. Though some researchers use indistinctly microbiome and microbiota, but by definition, they are two different things. The microbiota is a collection of all these organisms, and while microbiome is referring only to the genetic material. And this is other issue here, the metabolome. See, the bacteria that we have in our gut produce a lot of metabolites that influence either our brain or other organs, and this is called metabolome. And finally, we have the probiotics. So we have two other terms that we need to review. Probiotics are live microorganisms intended to restore the damaged intestinal flora. For example, yogurt is one of a good example. Another fermented foods like kimchi. I don't really how to pronounce the other ones, but these are the probiotics. A difference of what prebiotic means. Prebiotics is a food that will help us to grow a healthy bacteria in our gut. Okay. The number of bacteria in human cells. This has been a point of controversy during years, but it was based on the reason that 1 gram of feces contains 10 to the 11th bacteria, but it was just based on a final portion of somebody providing his feces, not a consistent measure over time. In 2016, in 2016, the number there is wrong. It's 2016. Milo and colleagues in Israel decided to get a more accurate approach on the measurement or estimate of the number of bacteria in the body and on the feces. We know that the colon has the largest number of bacteria. Well, we have in every gram of substance in the stomach, we have only 10 to 100 bacteria per gram. In the colon, we have 1 trillion per every gram of feces. So they took it wrongly, and they estimated the body to have the number of bacteria in the body to be in the order of 100 trillion. So they gave us a ratio of 10 to 1 at that time. And if you look into literature, lots of studies still have this number. However, this new estimate gives us a ratio of 1.3 to 1, which is roughly about 38 trillion of bacteria compared to 30 trillion of bacteria in the human body. And that's interesting to know, but however, this doesn't, take away the importance of the bacteria present in our intestines. So the other terms that we don't need to know is about the eubiosis and dysbiosis. As we can see here on the this picture here, we have a healthy microbiota, and in the center we have the unhealthy microbiota. So what is considered a healthy microbiota? A healthy microbiota is considered when there is a marked diversity of microbial populations. Roughly, it's about now we have about 5,000 species of microbes in our intestine. So the abundance of these microbes is what is considered healthy microbiota, considering the fact that when one population of microbes disappears, of good microbes disappear, an unhealthy type of microbes overgrow. So the other condition is, this is the eubiosis. Dysbiosis is when we start losing this diversity of microbes in our gut. So this is a condition marked with loss of diversity, and the causes are very, very different type of issues there. Like, for example, diets in high processed foods, lack of regular sleep, stress, infections, the use of antibiotics, especially in children. Amoxicillin, for example, will cause that. And chips, potato chips and all that kind of chips, because it's highly processed food. And lots of us, we give those to our children. So is there a chance to restore the unhealthy microbiota? Yes, it does, but it takes time. So we need to change diet. We use stress management techniques, exercise. Targeted supplementation refers to there are capsules now that provide with probiotics, and also the use of fermented foods like yogurt and etc. And the last thing we could, I have a friend, he's been donating his feces for another one that didn't have a good microbiota. So fecal transplant is a way of treatment nowadays. What else we have here? Okay, this thing is not moving. Let me see if we can. Okay, it does. Went slow. So what is the dysbiosis effect? Now that we know that what is dysbiosis, we understand that this effect of dysbiosis, dysbiosis will cause a tremendous damage to our body. If we look in the deep blue, we have anxiety, depression, schizophrenia, autism, Parkinson's disease. These are matter of research, and little by little is being proved that these are related to deranged or dysbiosis in our intestines. And since this affect the organs in the abdomen, in the intestines and stuff, we have colon cancer, irritable bowel syndrome, obesity and diabetes, Crohn's disease, ulcerative colitis that are related to this dysbiosis or derangement in the composition of our microbiota. Recently, just last year, a study has found that the prevalence of Prevotella copri, one of the Abel bacteria in our bodies, that is related to rheumatoid arthritis. So the focus of this research is going into finding ways to give a treatment to or eliminate this bacteria in our body or in the microbiota. So we have now what is called bacterial villages. Certain areas in our bodies have different populations of bacteria. Because of the difference in the predominance of certain type of bacteria, they call it bacterial villages. As we can see in this picture here or graphic, we have behind the ear a great population of bacteria. That's called the retroauricular zone. In the nostrils, with the predominance of a bacteria called Moracella catarrhalis, something like that. In the tongue, in the throat, in the axillary zone, in the large intestine, and the genital area. Those are the main bacterial villages, and we have to deal with. And so the next question we want to look into is the microbiome in human health and disease. This is this describes us what is going on here. We have an environment, geophysical environment. We have also a social environment. As you can see here, that's called exposome, because that will affect to those that are exposed to these environments will have its effects. One of its effects would be on the food we eat. But we can have a balanced diet. A balanced diet, according to this research, is decreased red meat consumption, increased fruit and vegetables, decreased methionine content, high variety of foods. And on the other hand, imbalanced diet. An imbalanced diet consists of excessive red meat consumption, low consumption of fruit and vegetables, higher methionine content, and low variety on our foods. So if we have a balanced diet, we can have a normative microbiome. It means the microbiome is the one who will regulate our health. And on the other hand, we will have an aberrant microbiome, which will push into the disease state. So if we have an aberrant microbiome, how disease will go? We will age more rapidly than our biological age. We will have this condition of metabolic morbidity. We will increase our BMI easily, and our NRF2 will be decreased, and inflammation will increase phosphate. The mitochondrial biogenesis will be depressed. That means our energy will be less, and there will be epigenetic dysregulation. And of course, it will affect the brain also. We'll have adverse cognitive effects we mentioned before, like, for example, Alzheimer's, and one of the other graphics. The health will be pushed by the normative microbiome. We will age normally. We will have a metabolic status under what's called the normal quality of metabolism. and BMI will be normal. Our NRF2 will increase. Inflammation and phosphate will decrease. So that's the relationship with the microbiome and the disease. Okay, let's move on. These people here are from a tribe called the Yanomamo in Venezuela. But also together, this is just one of the tribes that I picked for the purpose of this presentation. We have in other studies that were done in Namibia and other places of the world, where the microbiome of these people has been compared to the Western diet. The diets were compared in terms of the microbiome, how diverse is the microbiome in these tribes compared to our Western diet food eaters. So what's the difference? What was found in these groups? The thing is that these groups contain less protein. They have natural sugars, not too much fat. They don't use soap. They don't use hand sanitizers. What is significant of these studies are that the impact on their lives. If we look at the health of Westerners, and we compare to them, we see that they do not die from the same diseases that Westerners die. I mean, with the diet in the US. Could it be that the diet of these people or the microbiome that these people contains or holds the key to our health? So this is one of the questions that comes from this. If we look at this next graphic, we have the Western microbiome on the left side, and we have the non-Western microbiome on the right side. So which do you think has the more variety or more diversity of microbes? As you can see, on the right side, you can see a tremendous difference in diversity of microbes in their microbiota. And this is what non-Westerns are providing to our knowledge. And we think that this could greatly, the knowledge of this, of the difference in this microbiota would make a great difference in our health. So let's look at this. Another issue here, immigration to US. Let's look at this difference. When people is coming, let's say from Namibia, from these primitive societies, quotes, not primitive, but from native areas, if they come from these countries, we call this a pre-immigration status. As you can see there below them, you see the microbiota, very diverse microbiota. As they arrive to the US and start eating the food that we eat in the US, the good microbiota, the good microbes are disappearing. And if they remain as a long-term residents in the US, the microbiota is even worse. It's going to dysbiosis. And on the second generation, they have clearly changed their microbiota. And we have a microbiota that is in dysbiosis, which will lead us to disease. And as I said before, if the good bacteria is decreasing or depleted, like this, in this case, Prevotella, which is a fiber that degrades, it's enzymes and provides us with short chain fatty acids. And on the counterpart, we have the Bacteroides strains, which is a bad bacteria for us, that increases its number in the population. So when people migrate to the US, there is a loss of diversity in their microbiota, and they start losing the good bacteria, and they start growing a bad one. So here's another thing. This is another thing to consider, factors shaping the neonatal microbiota. As a child is born, it could be born by vaginal, it can have a vaginal delivery, or it can be born by C-section. So these are two different things, different ways to be born. But the difference is when a child is born through vaginal delivery, it acquires the microbes from the birth canal. In this case, for example, the acidophilus. If a child is born by C-section, none of the bacteria that the mother has will be acquired by the child. So what happens then? When a child is born by vaginal delivery and is breastfed, the breastfeeding will reinforce the bacteria that he has acquired from the mother, and by 2 to 3 years, will basically have a microbiota that will help him reach adulthood. So if a child is born by C-section, he will not have the bacteria and will have lots of problems. And it's going to be worse if he's not breastfed. So some hospitals, what they are doing is, as soon as the baby is born, they swab the vaginal area from the mother, and they spread it on the face of the baby, so they can acquire those microbes that will help him to develop a good microbiota. And recent research also has found that we can talk now about heritability. Great part of the bacteria comes from the mother, and this type of bacteria is usually shared among the members of the same family. If we have a family and compare to another one, most of this, the first family will have basically the same bacteria among all the members, and so will be the other family from their own family. So this is very important because that's the way it develops bacteria, and bacteria is important in the child. To have a healthy microbiota, because microbiota helps or trains the immune system to respond to external agents, to antigens that are unfamiliar with them. And also it has to do a lot. Recent study in University of California, San Francisco, they are studying the impact of a child without microbiota in developing asthma. Okay, now this is another very important thing that we need to consider during the study of microbiomes or microbiota, the brain-gut microbiome axis. What is this? The brain-gut axis is carried out via vagus nerve, the 10th cranial nerve, or the main nerve in the autonomous nervous system response. It's also done via immune system or via short chain fatty acids, as I mentioned just a while ago, and the tryptophan serotonin axis. So how does the brain respond to this? It increases appetite and energy, homeostasis. It has to do a lot with the stable emotional status and attitude, how we respond to stress, and it's also involved in learning and memory, but also how we respond to pain. And on the same token, the gut or the microbiota responds via the enteric nervous system, sending molecules that will influence the neurotransmitters like serotonin. Will also have to do with stress, anxiety, mood changes, behavior response to pain, nausea, vomiting, and this brain will influence the gut with increasing the motility, the secretions, the delivery of nutrients to the rest of the body, the balance between these groups of bacteria in the body, like we said, the bacterial villages, and also response to pain. So what do we have next? We continue with the brain-gut microbiome interactions. So what do we have here? How do we explain this? We have gut microbiota, we have the central nervous system, we have the intestine lining, and the neuroendocrine system that works in the intestines. So what we see here is that the gut microbiome, right here on your left side, interacts with the brain via microbe-derived neuroactive molecules. It releases molecules that will act on the brain system. The brain system will respond with releasing molecules of the neuronal that influence the neuronal, immune, and neuroendocrine system in the gut. But that is done via the vagus nerve, increasing motility, secretion, permeability, and will act on the microbiome or microbiota, modulating its behavior. The gut microbiota acts on the intestinal lining, also releasing molecules that will modulate the response of the intestine, and on its turn, the intestine will reply to the gut microbiota, will communicate with the gut microbiota to modify its behavior by releasing what we called gut-derived molecules, and thus we have a brain-gut microbiome system communicating each other constantly to benefit or to improve or to change or to modify our health. So this is why it's so important to know about this, so we can, look into the direction of what can we eat properly to maintain a balance in our microbiota. Okay. This is just a brief example of how the microbiota is also intervening in the modulation of our behavior. The serotonin signaling is implicated in the reward system. That is one of the hormones that make us happy. If serotonin is very low, we come into a state of depressed. That's why the psychiatrists use serotonin similars to treat depression. But we need to go to the cause of all this issue, and the research is finding that the gut is involved in this depressive state, because we know that serotonin, 90% of the serotonin is produced in the intestines, in what we call the enteroendocrine system. So it affects the production of serotonin. If there's enough dietary tryptophan, which is a precursor of serotonin, then we made it. We can have our emotions, our state of mind in check. So serotonin is released, it goes to the brain responds, and we can have a balanced state of mind. Our health is in good shape. So this is just briefly. There are other things that we could explain. This is interesting. Recently, they came up with 3 studies, one in India, Hong Kong, and UK, with similar results. And this is that the gut microbiome composition is altered completely during a COVID infection. And this is in comparison to non-COVID individuals. What is important, during the COVID infection, we talked a lot about cytokine storm, how it is done. The thing is that composition of the gut microbiota in patients with COVID has an elevated number of several inflammatory cytokines, chemokines, and blood markers of tissue damage. These things are really increased during the COVID because COVID damages a group of bacteria, a bacterial village composed by Faecalibacterium prausnitzii and Eubacterium rectale. These two bacteria or microorganisms have been depleted during during COVID infection. So there's not, since microbiota is involved in modulating the immune system, there is not a good response from the microbiota to these viruses. And the thing is that after the virus is cleared from the organism, this situation persists in the body. And that's how, and all these three studies have concluded about the similar results. Okay, I don't know if we need to have time for this. I don't think we don't need to go there, but we need to look into this. The worst enemies of the microbiota is primarily due to our unhealthy diet. I mentioned already what are those elements that affect the microbiota. So one interesting study says, the one that we've seen previously with the Indians, with the Yanomamo in Venezuela, they have lots of natural sugars. They don't have refined or highly processed foods. And the other thing is sedentarism, lack of exercise. Lots of us have so many important things that we don't take time to exercise. Other enemy for the microbiota is antibiotics, and we have a clear example with the chronic ulcerative colitis, where bacteria is depleted, and we have the abundance of Clostridium difficile, and we usually treat with antibiotics, which makes we cause more depletion. And also, I mentioned heritability, the type of bacteria that comes from the mother, and the environment, also the pollution that causes a tremendous damage to our microbiota. Okay, eating right. So we are talking, since we are talking about diet, how diet affects our microbiota, it's a question of asking, how can we eat right? We learned that eating right is associated with a healthy microbiota. So we need to know what kind of healthy foods we need to eat, what are the elements that we need to have in our food in order to be healthy. Because eating poorly affects us in the sense that we cannot get the nutrients that our body needs to maintain us in a healthy status. An unhealthy microbiota will be improper to break down the nutrients for proper absorption and nutrition. So these are major molecules that we need to have in our food. Tryptophan, because it's the precursor of serotonin. Serotonin will make us happy, will stabilize our mood. And also we need melatonin to sleep, have a good sleep. And we need tyrosine in our diet, because we need dopamine. It's derived from tyrosine, which regulates pleasure, regulates memory and motor skills. We need epinephrine to be ready to fight or to flight. But the continuous effect of epinephrine or adrenaline will give us a lot of problems. We need also the thyroid hormones to keep in check our metabolism, and also the melanin for the skin pigment. And this is one important thing that we need to have in our diet. It's the indole-3 lactic acid, or ILA, which is the precursor of IPA, or indole propionic acid, is one of the strongest antioxidants in the body, and also it's involved in the production of short-chain fatty acids that we need. These are the healthy foods that we need to have. The 4 groups: abundance of vegetables, fruits, nuts, and grains. This is interesting. What I find, I don't know if you have seen this. This is the periodic table of microbiome or friendly foods. This is proposed by the Prostate Cancer Institute. We have lots of good foods here. This is interesting. I just put it there just for information. Maybe you would like to have it. And there's a current research on the microbiota. The study is in the direction of the metabolites that are produced in by the good microbiota, and also the metabolites that are produced when the body is in dysbiosis, and also the factors that influence and balance the microbiome. And also the probiotics development target specific needs. For example, if your Prevotella is decreasing or has been depleted because of COVID, then you can have a capsule that contains Prevotella, so you can replenish your need of Prevotella. So those are the things that have been worked on, and there are some studies. Especially the University of California, San Francisco has an institute of microbiome. There are interesting studies there. I just want to mention it because to reaffirm what I said in the beginning, the difference of Western diets in the analysis of their blood. They found that there's there's abundance of alpha amylase in the blood of the natives, and this is understandable because they don't have highly refined foods. The alpha amylase processes complex carbohydrates in their diet, so we will not find like a monosucrose to enzymes that break down only simple carbohydrates, because that's what you have in refined highly processed foods. So we find in their diet alpha amylase for complex carbohydrates. The other thing in research was done in rats. They provided two groups of rats. One is both are depleted of microbiota, but both were given one of the groups were given microbiota coming from obese rats, and soon after they started gaining a lot of weight and became obese. And that's one of the ways that it's kind of proving that the effect of the changes in our microbiota have to do a lot with obesity. Other research groups done in humans haven't been able to prove how the microbiota affects results in obesity. So this research is wide, and we are invited to participate. It was just at the beginning of the last decade when the interest for the microbiome increased. We could say that in the beginning, the literature was purely descriptive. There wasn't a research established, like focusing on certain direction. It was just describing what was going on in the field, but nothing with major consequences to our health. So, due to the new technologies developed after the completion of the Human Genome Project, a lot of people decided to study other genomes. especially those of the bacteria found in our body. So microbiome research is focusing on functional consequences of altering the gut of microbiota, like I mentioned in the rats, for example. Given the rats microbiota that comes from an obese rats, these rats that didn't have a microbiota, they became obese. So we cannot ignore the excitement that the research on this field is not only promising, but we are seeing positive results. So understanding our microbiota and its relationship with the mind and body will help us find mechanisms and appropriate interventions to deal with chronic diseases and mental health issues that nowadays, not only is expensive, but it's ineffective. So if we can find a way to provide people with not enough funding to, just change their diet with the guided, intervention, I think we could do a lot better with that. I know it's not an easy task. It's like not everybody will believe all of this new way of lifestyle, but I think research is moving in the right direction. It's giving us another tool to deal with these difficult or diseases in our modern medicine cannot really achieve good results. So I'm excited about this new field. I wish it could get into real research on this, but thank you so much for your attention. It took me a good amount of time to find these results that I'm presenting to you. So thank you so much for your time. I hope it's been helpful to your understanding of what microbiotas or microbiomes are, how we could do better for our health.